Hey everybody, thanks for coming. It's my pleasure to introduce Kieran Samuk. Kieran and I go way back. We overlapped as undergraduates actually at the University of Toronto. One thing I like about Kieran is that he's got really diverse interests, which were sort of fostered, I think, in our Toronto environment and subsequent training. So he's actually now worked on with bumblebees as an undergraduate, and then a master's with spiders, and then a PhD. You may know Dolph Schluter's lab, big lab for stickleback evolutionary ecology research. So Kieran was involved in kind of moving that lab group towards the genomics side of things. And then he also went on to do a postdoc at Duke with Mohammed Noor, where he started working with Drosophila and studying recombination rate variation. And so Kieran has a really diverse kind of history and set of interests and contributions to the field. But today is going to focus on the evolution of recombination rate in, I think, both stickleback and Drosophila. So welcome, Kieran.
Excellent. Thank you so much. Yeah, thanks, Nathaniel. Thanks so much for the invite. I'm really happy to be here. I've been talking to a couple of people. I'm originally from Ontario, Canada. I've been living in Southern California. And I was afraid that I wasn't going to get to see snow. And I know you guys haven't had a lot of it this year, but as soon as I got off the plane, I was like, yes, snow. So yes, thank you all for your contributions to that. So yes, today I'm going to be talking about this topic here, the evolution and consequences of recombination rate variation. And I'll be talking about it in both these organisms. And I made this little slide here for a talk at some point, and I like to think that this is Drosophila pseudo-obscura here, and this is a three-spine stickleback, and this, I like to think of this Drosophila sort of having, like, you know, the scene in The Lion King where Simba is looking up at the clouds and Mufasa appears in the clouds, and he's like, "My son, you must take your kingdom," or whatever. This is kind of what I imagine going on in this picture. All right, anyway, so let's get rolling here. Oh, I broke it. There we go. Okay, so I'm an evolutionary geneticist. And so what that means is I'm interested in how genetic variation that we see out in the world gets filtered through the various processes of evolution to give rise to all the amazing diversity we see out in the natural world. Oops. And so the focus of my lab touches on various different topics related to that process. So here are a couple of things that we're working on. What are the genetic changes that underlie the formation of new species? And we're working on this in both sticklebacks and flies. We're also becoming increasingly interested in how structural variation in the genome contributes to adaptive evolution. And this is being enabled, as many of you all know, by all our cool new long-read sequencing technologies. And what I'll be talking about today is another big focus of the lab, which is what role does recombination play in evolution, and does it itself evolve? Okay. So when we think about where new genetic variation comes from, so genetic variation is the raw material of evolution, where does it come from? One big source is new mutations. And so you know, if you talk to Nathaniel, you know that these PowerPoint lightning bolts often arise in genomes and cause little mutations to occur. And that is the ultimate source of new alleles in populations. Obviously, it's a little bit more complicated than that. From the perspective of a population, another major source of new allele variation is gene flow. Alleles from other populations moving into your local population. Another source of genetic variation, not usually one we kind of think of in the same way because it doesn't necessarily change allele frequencies, but another big source of genetic variation is recombination. So that is the mixing up of genomes between an individual to give rise to sort of a mixture of gametes with variable haplotypes. So recombination does not change allele frequencies. So it's not really a force of evolution if we define evolution as a change in allele frequencies. Nevertheless, it does play a really extremely important role, I think, in evolution because it creates new combinations of alleles, that is to say haplotypes. So, you know, if we think of, for example, this cross here between two parents giving rise to this F1, all of these new haplotypes here have not existed before, don't exist in these grandparents. So that's a source of new genetic variation. So what I'm going to talk about today, I'm going to outline a viewpoint here that recombination rate plays a key role in shaping many of the evolutionary processes that I'm interested in, many in the field are interested in, and also that it itself is a phenotype that varies and evolves in natural populations. And so, this latter component is something that, for example, Brett Pazer has worked on a lot here, and we're hoping to make some contributions to that question as well. Okay, so quick outline of the talk. I'll start by giving an introduction to recombination. And I know that this is a genetics department or a genomics themed talk, but I just want to make sure we're all on the same page about what we mean by recombination. I'll then give one vignette of a study we did looking at how variation in recombination rate affects patterns of divergence in the genome and might influence the way that evolution unfolds in populations. I'll then give another quick vignette on some work I did during my postdoc on the quantitative genetics of recombination rate variation in flies. And this study is kind of the theme that my lab is now sort of centered around. And then I'll just very briefly talk about some of the future work that we're working on now in my lab at UC Riverside. Okay, as many of you are very well aware, our genome is, many genomes of organisms are organized into discrete units called chromosomes. So here's a human karyotype. We're all very familiar with that. We can sort of symbolically represent this, showing the two different copies of each chromosome here. Throughout the talk, I'll also be kind of using this type of notation where we tag each of the ancestries of or origins of each one of those copies. So we might say this individual gets one copy of each chromosome from his mom and one from his dad in this particular case. Okay, so recombination in general is the exchange of genetic material between homologous chromosomes. So the form of recombination I'm going to be really focusing on today is crossing over. There's other types of recombination. Gene conversion is another one. We won't be talking about those today, but I'll basically be using sort of crossing over and recombination synonymously here. So just to remind you, here's our chromosomes during meiosis. They've undergone doubling, so we have our two pairs of homologous chromosomes here. During meiosis, crossing over can occur where one arm of one of the sister chromatids of the two homologous chromosomes exchanges material with the other, ultimately giving rise to 4 different haploid gametes with different combinations of genomes. So this is that, for example, here we have 2 brand new haplotypes that have not existed in the parental genome before. Okay, so that's recombination. And once again, using that synonymously with crossing over. What about recombination rate? Well, recombination rate is just the average number of crossovers per generation in some interval. So say we have a chromosome, and we have our diploid parent here with its 2 copies of its genome or chromosome tagged. That individual, through that process during meiosis, might give rise to many different possible recombined haploid gametes. If we wanted to estimate a rate of recombination for this interval, this chromosome, we could count up all the crossovers that have occurred in the offspring and take an average of that. Oh, my laser pointer just died. That's unfortunate. Take an average of that and get a rate for that particular chromosome. So that's recombination rate. Okay, so I am an evolutionary biologist, and one of the things that I'm interested in, or really fascinates me about recombination rate, is that it dictates the strength of linkage, kind of by definition, but it also has a really important role in dictating what we can kind of call the granularity of genetic variation in a population. So if we imagined this recombination process unfolding in a particular chromosome, we could maybe envision a case of low recombination rate on a chromosome and maybe high recombination rate. When recombination rate is low, we may end up with just 1 or 2 maybe crossovers on a particular chromosome. And so we have very long stretches of sort of ancestry where we look at each of these haplotypes. We just have one kind of breakpoint separating these, but most of these alleles are all in association with the alleles they were before. We can contrast that to a case of higher recombination rate, where we could have maybe double, triple, quadruple crossovers or something like this. And we can see that the genome is much more mixed up. And so kind of one way to think about this is when recombination rate is low, genetic variation is kind of contained in like big boulders that are all kind of stuck together. Whereas when recombination rate is high, allelic variation is much more granular, sort of more like grains of sand in terms of how they will be seen by the forces of evolution. Okay. So another really fascinating thing about recombination rate, we're still learning a lot about this, is that it varies at almost all biological scales that we look at. So this is one you probably are familiar with. A recombination rate can vary within and among chromosomes. So this is some work from Josep Comaron. This is the second chromosome Drosophila melanogaster, the two arms tagged there. On the y-axis, we have crossover rate. On the x-axis, we have coordinates relative to the centromere. And you can see that this is a wiggly line. There's lots of variation here. There's variation at different scales as well. For example, we could see around the centromere, recombination is lower, but there's also all kinds of interesting local variation in recombination rates. So this is interesting. So, variation is the stuff of evolution, and here we have one source. Variation, recombination can also vary within and between individuals. Here's some data from corn. So what we have here are recombination rates, a sort of heat map here. So cells that have warmer colors have higher average rates, cells that have cooler color are lower. And each of these rows here is a different mapping population of corn. And each column is a different chromosome. So, just kind of take home message here. For example, if we look at this mapping population here, which is from a single genotype, you can see that genome-wide, it seems to have much lower recombination rates than other such populations. We can also see chromosome level variations. Something's going on this chromosome, for example, that makes it much lower than others. So once again, we're seeing lots of variation in recombination rate. And so, yes, as an evolutionary biologist, this is very exciting to me because this suggests that the raw material of evolution is there. And finally, we were talking to Brad about this figure earlier. Finally, we know that recombination rate varies greatly across the tree of life. So this is kind of going out into the macro scale now. So this is a figure from this review by Jessica Stapley et al. And what we have here are all different types of taxa on the x-axis. On the y-axis, we have recombination in a log scale. So there's enormous variation in recombination rates among taxa. Some of this is explained by chromosome number, but even after accounting for that, there's substantial variation in recombination rates among taxa. And so once again, this is very exciting, lots of variation. So, as an evolutionary biologist, immediately the question I wanted the answer to is why? Why is there all this variation? Whence cometh all of this recombination variation? And getting a handle on that requires measuring it, and ideally in multiple individuals and multiple different scales. And one of the frustrating things about studying this, we have this like tantalizing cool variation. We want to go out there and understand it, but measuring recombination rate is extremely annoying. And any of you who have done this or made a genetic map know that this is annoying. The sort of traditional way of doing it, going back to like Sturtevant and Thomas Hunt Morgan, is we make a genetic map. We make crosses, we track, recombination frequencies and markers, and then we build some kind of linkage map on the basis of that. And that is very laborious. It's very limited in the number of organisms you could do it in. So there's big limitations to the methodology here. Okay. Luckily, work's being done here, and there's all kinds of exciting new recent technologies that are providing many new ways to study recombination rate. So one approach to this is to use population genetics. And so, as you all know, we now have tons of population genetic sequencing data and all kinds of different model and non-model organisms. And we can use model-based approaches, for example, to get estimates of recombination rate. Recently, and this is something my lab is working on, there are also ways to do direct gamete sequencing. So, for example, single-cell sequencing of sperm, and using those as the individuals to identify haplotypes with crossovers and get recombination estimates in that way. So that sort of shortcuts having to actually make a genetic map and genotype offspring. You just genotype the gametes directly. There's also a lot of really interesting new tools for the transgenic manipulation of recombination rates. This is stuff like inducing double-strand breaks using CRISPR, or manipulating genome structure to make and destroy chromosomal inversions, all those sorts of cool things. So lots of new tools coming online. And so this is exciting for me because, thinking about all that variation out there, wanting to explain it, understanding the both causes and consequences of this variation, I think is now becoming within our grasp. And so what I'm interested in is establishing the links between how the variation and recombination that exists out there influences the forces of evolution, and how those same forces kind of feed back and can shape variation and recombination rate itself. Okay. So this first vignette is going to be focusing more on those consequences. We're going to be taking some variation in recombination rate in an organism and trying to understand what consequences does that have for its evolution. So this is some work that I did with a group of folks from UBC, and this was published in Molecular Ecology a couple of years ago. Okay, so the background, and we kind of take this as given now, but adaptation to new environments, as I'm sure many of you know, often occurs in the presence of gene flow. So most organisms are not completely isolated from all other populations of the same organism, and exchange genes between populations experiencing different selective pressures. And so here's a sort of schematic of what this scenario might look like. So this is the scenario of divergent selection with gene flow. So we're sort of have a little phylogenetic tree here. So time is on the x-axis here, proceeding forward in time. So we have some ancestral population that say splits to inhabit two new environments. Within each one of those environments, selection is sort of building up sets of alleles that are locally favored. It's driving alleles that that work in that population to higher frequency from that ancestral state. So we can kind of envision these little blue mutations giving us this sort of blue haplotype at the end. That same process is also occurring in that other population. So, the alleles that work in the red population are also building up. So what happens when there's gene flow? So let's imagine we have some migrant haplotype here coming from the red population. If it migrates to the blue population and does not die immediately, it can interbreed with the local types. And so what this means is that recombination will sort of start mixing up those two genomes. So start mixing up those red and blue genomes. And if those sets of locally adapted alleles, those blue alleles, are all being intermixed with these maladaptive red alleles. And what we were interested in for this project is what role does variation in recombination rate along a chromosome play in mediating this process, and how this all unfolds. Okay, so the other thing that motivated a lot of this work was a body of theoretical work that basically has this prediction. Regions of the genome that have low recombination should be sort of more resistant to maladaptive gene flow. And there are a number of different authors who have explored this idea. This idea is also very analogous to the work on the theoretical work on the evolution of chromosomal inversions. But so I'm going to go through some of this theory, but instead of giving you the math, I'm going to do it all in fun pictures instead. Nathaniel's laughing at me, but here we go. So here are the basic additions of the model. So we have divergent selection with gene flow. That's the situation we talked about before. So we have some migrant population that is migrating into another local population where those migrant alleles are disfavored. And the other kind of implicit assumption of this idea is that there are many genes that underlie local adaptation. That is to say, local adaptation has a polygenic genetic architecture. And so we can kind of just imagine that most of the genome is contributing in some way to local adaptation. So those are the kind of two main assumptions that we're talking about here. Okay, so how does this kind of play out when we think about how a migrant type might intermate with a local type? So if we kind of do a cross between these two, we end up with the F1 hybrid individual. If those F1 hybrids persist, they might intercross with other F1 hybrids or other local types in some combination. But if we kind of imagine the outcome of several generations, we have some kind of mixed up, chopped up genome. So how chopped up and kind of where it's mixed up is going to be dictated by recombination rate. So if we kind of imagine the rate of recombination might look like this, where we have some low point on the chromosome and it's kind of higher at the tips. This is fairly typical, whoops, higher at the tips here. We might expect sort of more mixing to be happening at the edges of the chromosome and less in the center. This is just a natural consequence of variation where crossovers occur. Okay, so what does this have to do with selection? So the kind of key here and the key insight of these models were, okay, if we kind of think about these little chunks here, these regions in isolation, in aggregate, contain fewer maladaptive alleles. Like, let's say that, each little length of the chromosome contains 10 maladaptive alleles. This big chunk here is going to have a ton of those, whereas these little chunks are going to contain far fewer. And so if you have a big linked block all full of bad alleles, that thing is going to have, in theoretical terms, a low selective coefficient, and selection will have an easy time purging that from the population. So these things will be readily eliminated because they're unfit. In contrast, if we had just this little nub in here, this might be, below the selective coefficient to even be affected by selection, and would just kind of drift around. And these smaller chunks will take much longer to eliminate if they're eliminated at all. So this idea, if you kind of think it through, and luckily some smart person did, we kind of might end up with a kind of situation like this after gene flow, where the sort of core of the chromosome has effectively resisted recombination by being more efficient at purging that those maladaptive alleles, whereas the periphery of the chromosome is going to kind of be more less resistant and maintain a lot more migrate alleles. So what this is setting up is a relationship between the efficacy of selection at purging migrant variation, and if we then compare, for example, genetic differences between our blue and red populations, what we would expect here, if this model is correct, is that in the center, there should be mostly different alleles, and there should be a lot more sharing of alleles at the periphery of the chromosome. And so this leads to this key prediction in terms of testing these models. When we have the co-occurrence of divergent selection and gene flow, we should expect genetic differences between populations to be greatest in regions of low recombination. I hope that did the math justice. Okay, so that's all sounds great. How do we test this idea? We want to bring data to bear on this idea. So once again, here's that prediction. When divergent selection and gene flow co-occur, these genetic differences should be greatest in regions of low recombination. So our idea here was to focus on this requirement and find counterexamples to this that would kind of support the opposite. And so, yeah, our idea was to compare genomic patterns of differentiation between populations paired that actually vary in the presence or absence of gene flow and divergent selection. And so we kind of, I know we as biologists, we love 2x2 tables, and I'm no exception. So if we kind of imagine where we expect to see this pattern, it's the coincidence of divergent selection and gene flow is really where we're expecting to see it. We're expecting to see some other baseline pattern elsewhere. So both of those things need to be present for this theory to hold. So three-spine sticklebacks, which are this little freshwater fish here, are a really great system for that, for testing this question, because we basically have many, many independent populations that vary in the presence of absence of divergent selection and gene. And so we could do this. We could fill out our little 2x2 table. They also have, for vertebrate, quite good empirical recombination estimates, high quality genomic resources, and this replicated divergence that gives us sampling replication. And so what we did was we basically at the time collected all the cross-population stickleback genetic data we could get our hands on, variety of different technologies, all kinds of different interesting divergence scenarios. But what we did was we took all that genomic data, and because of all the amazing natural history information we had on sticklebacks, we were able to plug those all into our little 2x2 table here. And so this is the number of pairs of each one of these scenarios that we came up with. So many different examples here. This one's a little depauperate because if you think about what that is, those would be like two populations of a very similar thing that differ in no discernible selective traits. It's not usually something that evolutionary biologists study. We usually study stuff that's different. But we did find lots of 31 of those. Okay, so the basic idea here is we take these populations, we want to test this idea. And so we wanted to start by examining patterns of genetic differentiation. And so one way to do this is just with good old FST. So this is just a measure of genetic differentiation. Many of you are quite aware of it. So we could use just FST quantitatively, just as an index of genetic differences. We can also use, for example, like outlier density or something like that to sort of tag regions of high genetic differentiation. And that might be one way to approach this. I will say that we basically, there's, kind of fancier, schmancier ways to do this nowadays. You could use more ancestry-based approaches versus just, FST, but you basically get the same answer. Okay, so, here's kind of one way that we might use FST to test this. We might basically expect higher FST and more outliers in this part of the genome relative to the periphery. So more FST outliers in regions of low recombination, fewer FST outliers in regions of high recombination. Okay, so here's some actual data from 4 different pairs. So here's our 2x2 table. And what I've just plotted here is just FST. So it varies between 0 and 1. And we have position on a particular stickleback chromosome here. So a couple patterns emerge immediately here. So first of all, in our kind of expected cell here, we have this sort of hump-shaped pattern, whereas we sort of have a more flat profile for FST in the other examples. So FST is obviously going to be lower here overall because we have gene flow and no divergent selection. So that all kind of fits our expectations. This hump-shaped pattern is a little weird though. So what happens when we map on recombination rate? So this is a smooth of the actual recombination rate for the stickleback chromosome. So what you can see here is these, the area of increased FST coincides with the region of low recombination on the stickleback chromosome. So this is just looking at it on one chromosome. If we got to zoom out genome-wide, here's that same data across all the chromosomes. And we see this pattern more or less on most of the chromosomes, not all of them, but most of them. So here's those same 4 categories and recombination on the bottom. And as part of the lead up to this project, people had started doing PopGen on the sticklebacks, and someone had noticed this sort of hump-shaped pattern across the genome, now a collaborator of mine. And so this is Marius Rossi's, one of his papers, is a supplemental figure from this paper. And Marius called this the Toblerone of speciation. So Marius is Swiss, and he sees the world through the lens of chocolate, and so there you go. But that was one of the first times someone had shown that. Okay, so that's cool to look at pictures like that. We wanted to kind of summarize that pattern across the genome. So we came up with basically an index for this, for the bias for divergence in regions of low recombination. And we'll go into the math a bit, but basically the index is 0 when we have a flat profile, and it becomes increasingly negative as FST outliers are localized in regions of low recombination. So we could use that to sort of summarize this pattern genome-wide. And so if we do that, if we compute that summary for all 4 of those different categories, we end up with something that looks like this. So here are our 4 categories. So we have selection with gene flow here. That's the one we're expecting to see the pattern. And then we have our 3 other cases here. So you can see immediately the pattern jumps out that there's quite a bit of variation in the data. But on average, we see this pattern of more bias in regions of low recombination when selection and gene flow co-occur. So this is exactly what we're expecting. And so just to situate, link back to the data, that particular data point there is that chromosome I showed you earlier, or that comparison I showed you earlier. So there are ones that are even more extreme. Okay, so this is great. This is what we basically expect to see. So these comparisons, these divergence patterns support this idea. And we end up with these kind of specific profiles. So that's great. Big check mark there. So why is this interesting? So a couple of reasons for me as an evolutionary biologist why I find this interesting. First of all, one implication may be that the rate of recombination interacts with gene flow to shape where genetic differences between populations can evolve. Effectively, what this is saying is that because of the linkage relationships among loci, regions of low recombination, genes in regions of low recombination are more likely to contribute to genetic differences in the presence of gene flow. Whereas if there's no gene flow, there's sort of more of the chromosome open to evolve genetic differences. So this might actually kind of bias what genes are involved in adaptation. Another, remember we were talking about the original assumptions of the model. This also basically supports this idea that adaptation is polygenic. If adaptation isn't polygenic, we shouldn't basically see this pattern. If it's all driven by, a couple loci or something, we should not see this pattern. And so this is also consistent with the polygenic architecture of adaptation in sticklebacks. Okay. So, how am I doing for time here? Okay, so that wraps up that section on thinking about consequences, how recombination sort of shapes the evolutionary process. I now want to briefly turn to this project here, which has sort of become touchstone for me in our lab on the quantitative genetics of recombination rate. So this is work that I did during my postdoc in Mohammed Noor's lab, and all these folks contributed to it. So this is looking at how natural selection might shape variation on recombination rate in Drosophila pseudoobscura. Okay, so just to remind you guys, call back to this figure here. We know that recombination rate varies at all these interesting scales, but we have a really poor understanding of the evolutionary forces leading to this variation, as well as the genetic basis of those differences, especially in natural populations. So we wanted to kind of ask three questions here. How does recombination rate vary within and between populations? Once again, that variation is the raw material of evolution. How much raw material of, recombination rate variation is there out there? We were also interested in making some progress on this question of what are the genetic changes? What are the mutations that underlie these differences that we're seeing out in the world? And finally, the kind of money question is, are those differences in recombination rate that we see in some way driven by selection or something like drift? Okay, so this work was done on natural populations of Drosophila pseudoobscura. There it is. It's a native species endemic to the southwestern United States and northern Mexico. It probably looks like just a fly to many of you, but it's not just a fly to me. Okay, so we chose two different populations to work on for this. So one is from this place called Madeira Canyon, Arizona, and the other is from American Fork Canyon, Utah. So these differ quite a lot ecologically. One is a sort of subalpine forested area, whereas one is a more sort of scrubby chaparral desert sort of area. So when I first started working on this project, I remember looking at this picture and thinking that this looked like a sort of alien moonscape to me. And now that it looks like that out my window in Riverside. So yeah, anyway. Okay, so what we did was we collected wild flies from both of those populations. And then we created inbred lines isolated from the natural population. Once we had those inbred lines, we then went in and created multiple mapping populations for each of the source populations. And here's the basic crossing design. So we had our inbred lines. Those are the focal isolines here. And so MC is Madeira Canyon, AFC is American Fork Canyon. So we crossed the male of our inbred line to a tester line, which is just the reference genome line, to then generate an F1 individual. And then we crossed that F1 individual back to another tester line to generate a bunch of recombinant offspring. And we're basically measuring the recombination of the F1s here. So you might immediately be wondering, well, why didn't you just measure it in the inbred lines? And the answer is, if you want to make a genetic map, you need genetic variation to have markers for. You can't use traditional genetic methods to measure recombination in a totally inbred line. So that's why we did this. So this gives us a little bit of variation. So the idea here is our recombinations kind of occurring here. And basically what we're doing is subtracting out one of these tester line genomes here. So this individual isn't really contributing anything. We just remove that whole kind of blue chromosome, and we get all our little recombinant possible haplotypes. And so in terms of how this is actually determined when we sequence it, we designed a bunch of markers to be able to track ancestors here, such that we could find crossovers just by finding switches in homozygosity in the markers. So we chose markers that behaved like this. So we could, in our sequencing data, basically just find places where it flipped from homozygous reference to heterozygous alternate. And the way that we did this, the actual sequencing, was we used amplicon sequencing. So we found a bunch of SNPs that behaved in that way. And then we used this method called GT-seq. And so it sounds insane, but the way that this method works is we developed about 500 primer pairs and do an amplification in a single PCR reaction with 500 primer pairs. It sounds like something that should not work, but it absolutely does work. And it works really, really well. And so I highly recommend you guys check this out if you're interested in targeted sequencing and don't want to develop a SNP chip or something. This does a great job. You have to fiddle with the primer pairs to get them to all behave, but it's worth it. So that's how we developed our SNP panel. Okay, so let's get to this first question. How does recombination rate vary within and between populations? Okay, so just to give you a sense of what the data looks like, here are some genotypes I just pulled out from our sequencing population. So each column here is a different individual, and we basically have position on chromosome on the y-axis here. So we could classify individuals based on the number of crossovers that were detected in their genotypes. So we could have cases where there's 0 crossovers, where they kind of have a completely solid haplotype, 1 crossover where we have 1 switch from blue to red, 2, 3, 4, and so on and so forth. And so this is how we actually quantify the crossovers. And the black dots are just missing data. Okay, so here's the first thing that we found. A large range of variation in recombination rate among our inbred lines. And so here we have crossovers per genome on the y-axis, and here are all our different inbred line tags here. So you can see that there's variation, lots of variation, actually. If we kind of collapse that down into the 2 populations, we find that Madeira Canyon, which is the sort of desert population, had about a 12% lower recombination rate genome-wide. Okay, so the next thing we did was we were curious if these differences in recombination rate were specific to particular regions of the genome, or was this a sort of genome-wide thing? And so what we have here are the different sort of recombination landscapes across the Drosophila pseudoobscura genome. And so here it is up here in red, we have American Fork Canyon and Madeira Canyon. And you can kind of squint to that for a bit and look at it. But basically, there aren't any big differences in the recombination landscape, or none that were statistically significant. And so they're nearly identical landscapes. And what is different is basically in the populations that differ, it's kind of either bumped up or bumped down, the whole thing. So this seems to be variation in genome-wide recombination rate. Another way to look at that is just correlate crossover rate between different chromosomes. So basically what this is saying here, we have crossover rate on one chromosome, crossover rate on another. So if you have a high crossover rate on chromosome 3, you also have a high crossover rate on chromosome X. So, and this holds true for all the different chromosomes. So this is, again, just evidence for genetic variation for genome-wide recombination rates. Okay, so that gave us a little handle on how it varies within and between populations. What about this? What are the genetic changes that we can identify underlying differences in recombination rate? So we were a bit constrained in what we could do here, but we kind of looked at 2 different things. The first was looking at a panel of candidate genes, meiosis genes that had been annotated in Drosophila. And we also looked at the influence of structural variation. Okay, so we first asked if among line variation or recombination rate was in some way associated with genetic variation in any of these candidate genes. So we pulled out candidate genes from the literature. We then whole genome sequenced all of our lines. And then we basically just asked, is genotype associated with recombination rate? Okay, so we found 3 candidate non-synonymous mutations in meiosis-related genes. Here they are. So each one of these little boxes just shows the 2 possible genotypes. Remember, these are inbred lines. There's no heterozygotes. So on the y-axis here, we have the mean crossovers chromosome arm for that genotype and the 2 different genotypes. And so here's one particular residue in this gene, ASP. And you can see that if you have one form, you have lower recombination rate, the other you have higher. We also found two residues in the My41 gene that also showed associations with recombination rate. The major limitation here is because we couldn't do something like a cross or a GWAS, we can't really disentangle linkage relationships. So we're not sure that we're nailing the causative mutations here. What's interesting is from My41, other work, this is from Jeff Sikowski's lab at UNC, null mutants, so if you knock out or knock down the expression of My41, these show reduced crossover rates relative to wild type. So this gene is a pretty good candidate for a recombination modifier. So yeah, here's the recombination profile for wild type, and here it is for My41. Okay, so that's candidate genes. What about structural variation? This is something that people often ask about. So basically the idea here is how structural variation might influence or interface with recombination rate is if you kind of think of the concept of structural heterozygosity. So that's where your two genome copies vary in structure. So it could be the presence or absence of whole segments of chromosome or copy number variation or something like that. But because these things are trying to pair up during meiosis, if you have regions that are completely missing, you might imagine those things might interfere with recombination rate. So less opportunity for crossovers might mean lower recombination. And there's some evidence for this in different, for example, in Arabidopsis, there's lots of great evidence for this. So what we did was we basically went through and quantified recombination rate differences in windows along the genome. And we were looking at the difference between the inbred line and the tester line. And we basically asked, is a size difference just in terms of base pairs of a particular region in any way between the inbred and tester line, any way related to recombination rate? And we called structural variance using a variety of different methods. And we basically didn't find a relationship. So none of the variation we saw seemed to correlate with differences in structure. And this is consistent with, again, the changes in the recombination not being due to like landscape differences in recombination and more of a genome-wide thing. So it doesn't seem to be a strong influence of structural variation. Okay. How am I doing for time? What's my actual endpoint? Okay, good. Excellent. Okay, so this last question, are population differences in recombination rate driven by natural selection? So we use this approach called QST-FST. And basically what this is a method for, this method is designed for answering this question. Are population differences in some quantitative trait greater than expected under a model of neutrality, neutral evolution? And so the idea here is we build some distribution of FST between the populations using genetic markers that serves as a sort of null distribution of what we might expect allele frequency differences to look like between populations. We then compute an analogous measure of called QST that relates quantitative genetic variation within and between populations. And so it's basically a quantitative genetic version of FST. So the trick here is we're placing the variance in a quantitative trait on the same scale as FST. So that allows us to compare this QST value to the FST distribution. And basically, it's basically like an FST outlier analysis. We're looking for things with high QST relative to some null distribution. So we're looking for this sort of outer tail, and those are the things should be enriched for things under selection. So we went out and did RAD-seq to generate our FST distribution, and this revealed really, really low genetic divergence between these populations. So here's the FST distribution. This is Baron Cochran's FST. So there's a little bit more to the method, but basically the idea is we can kind of imagine some cutoff here where we were saying this stuff is sort of an outlier, and these extreme values are not really expected under neutrality. And what we found is if we compute that QST value for our recombination rate estimates among our lines, it's way, 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 way higher than we would expect under neutrality. So it's up here around 0.02. So it's way out in the little tippy tail here. And as you might imagine, that's not expected under neutrality. So this basically suggests that recombination rate has been in some way shaped by natural selection. Okay, so just to summarize, we found ample genetic variation for recombination rate. We have some preliminary data connecting those to mutations, and we think that we've identified a case where we have evidence of natural selection driving differences in recombination rate. Okay, so what I showed today was that recombination rate variation shaped for evolution can occur in the genome. I also showed that recombination rate variation is itself potentially under selection in natural populations. So maybe what I'll do is end there if I don't. Okay. So yeah, once again, we have this connection between recombination rate shaping evolution, and evolution may be feeding back and shaping recombination rate variation. I'll just very briefly now talk about the research that I'm doing in my lab. So I used to have this slide in my job talk, and it said future research, but this is now current research in my lab. So yeah. So basically the idea here that we're kind of following through on both of these lines that I talked about today. And we're trying to bring to bear these new technologies in order to study recombination rate in this sort of genotype phenotype fitness framework that some of you might be familiar with. So we're interested in how, identifying the genetic basis of this variation. We're interested in studying the population genetics of recombination modifiers in natural populations. And really, we're very, very interested in identifying what the potential source of selection might be on recombination rate in the wild. So I'll just give a quick vignette on each of my students' current work. So my student Gina, so she's working, among other things, on exploring natural variation of recombination rate in populations of stickleback. So we're basically extending this approach we used in the flies to stickleback, and specifically making use of this sperm sequencing technology to save our sanity from doing all those crosses we had to do with the flies. My student Mikhail is working on a kind of completely different angle, is thinking about how we might use this information on recombination modifiers from a population genetic perspective to accelerate agricultural breeding programs and hopefully save time in breeding. My student Alex is also interested in looking at the role of recombination suppression in the evolution of reproductive isolation. And this is particularly in populations of three-spine stickleback as well. Okay, thank you all for listening, and I'm happy to take questions. Yeah, right there.
Super political stuff. I have two questions, and they're somewhat related, but so if I'm not mistaken, the two populations or the sounds that you had which were both allopathic from Switzerland, right?
That's right, yes.
Do you have any interest or have looked at all populations that are sympatric? Because there's some suggestion, if there's a population that they can potentially hybrid with, that would be detrimental to, you might suspect that there's some importance of the combination rate being selected. Is that?
Yeah, that's a very interesting idea. So we haven't looked at that in the flies. Oh yeah, sorry, just to repeat with the Zoomers, what the question was, the populations that we studied of pseudo-obscura were allopatric from a closely related species that is known to exchange genes with it. The question was, did we look at those populations? And the answer is no. But my student Gina is actually doing that exact idea in sticklebacks. So she's studying populations that are sympatric with a sort of divergent type, and also ones that are allopatric. And the idea is exactly what you were saying, is the expectation is maybe recombination rate should evolve to be lower in the presence of maladaptive genes. Yeah. So does that answer your question?
Yeah. And then I was also just curious why that in pseudo-obscura rather than like lining acid.
Yeah, it's a good question. So one is my postdoc advisor Mohammed is a pseudo-obscura enjoyer professionally. And I think the other problem was one of the things that we specifically did not want to have monkeying around the data were inversions. And so both of these populations that we chose were basically homozygous for the same chromosomal arrangements. So that whole thing we could just sort of skirt by. However, I think in real populations, that's actually probably an important modulator for recombination rate. But I think that was the reason that was chosen. Sorry. Yes.
on the land, it's just one of the things you said that caught me off guard is that in your inlet lines, you don't see any headers past them. Does Obscura degrade that well? As compared to the melanogaster?
Yeah, so that was just for those particular genes, sorry,
Oh.
not genome level. Yeah, there's still heterozygosity. There was only like 20 or 30 generations of inbreeding. So there's still definitely residual heterozygosity, but in those particular genes, we didn't find it.
So this is the fact that you have reduced recombination rate in one species in the face of gene flow suggests that there's asymmetry, that the gene flow is primarily in the direction of the species with reduced recombination rate, because that would drive selection pressure to keep co-adaptive groups of genes together. And the fact that you're not seeing a similar reduced recombination rate in the other species means that there's probably no introduction from the other.
That might be true. Yeah, there are a few examples where we've actually been able to establish that connection specifically between gene flow and reduction of recombination, statistically speaking. But that would be what we would expect, I think.
So the system you were looking at, is there any evidence that there's asymmetry in gene flow?
In the sticklebacks or in the there's definitely asymmetrical gene flow in certain stickleback populations, and it like varies depending on the population of interest. In the sort of species pairs that were the ones that had the most extreme example of this low recombination bias, gene flow seems to be about equal.
You showed a very striking figure comparing the flies from the two environments where you looked at the recombination rate as a function of gene position. And it almost looked like they could be superimposable, except you mentioned there was a shift at the
That's right, yeah.
baseline. I was trying to figure out how you could possibly get such reproducible data from or reproducible patterns. And I'm just wondering, is there a possibility that it could be related to the way you're doing the sequencing, the upfront sequencing, that somehow the tool that you're using is just so, well, it's the same tool,
Right.
It's just on two different individuals.
Yeah.
You mentioned in the upfront sequencing that you had to do some fine tuning.
Yeah.
And I'm wondering if you looked at the sensitivity of these sorts of patterns to particular amplicons.
Yeah, so we didn't really, yeah, that's an interesting idea. So we didn't actually fully deploy the amplicon sequencing until it was optimized outside of the context of this. So we were basically figuring that out. I will say that there are, for example, some little cases like, for example, if you look here at this little skyscraper thing, it's a little bit different over here. So it's not like we completely recapitulated it. So there are some fine scale differences in the landscape, but most of the variation is explained by shifting the whole thing up or down. But the question in terms of how the sequencing approach might affect this, yeah, it's difficult to know. I guess one way would just be do like whole genome sequencing or something of the mapping populations. But yeah, it's something that we weren't really able to look at.
Thank you.
Thank you. Yeah.
Something that may help interpretation of this figure, is it reasonable to interpret the genomic location of increases or decreases in the rate as the location of an amplicon?
Yeah, so this is kind of like smooth. We're sort of connecting the dots between markers here. And this is also kind of, you can't quite see where the amplicons are because this is more based on the crossover frequency. So yeah, this is derived from basically those haplotype pictures that I was showing earlier, translating those into this sort of stair-steppy landscape.
Oh, I see. So the coarse grain is based on the number of samples.
Yeah, basically, yeah.
Question is, I don't speak technology, so I don't know how much your questions will detail work.
No problem.
But I was wondering if you know how much the different environments can affect transposon activities and how this could influence, your final result.
Right. So the correlation between like TEs and recombination or something like this.
The different environments could affect at a genome level.
Yeah, no, it's a good question. I mean, I'm not sure. So they do differ in temperature, that's a very obvious environmental axis. They differ, and one's hot and one's cold, basically. And yeah, I'm not sure if there is, a difference in TE activity as a function of temperature? That is probably something that somebody knows.
Don't know.
Yeah, I don't know, I'm not sure. But we actually did, to your question, we did actually look at differences in TE content among all of our lines. I didn't say that here. But that was one of the other things that we looked at within the sort of structural variation thing. And there weren't really significant differences in TE copy number variation or different TE families between the two populations. So that's sort of indirect evidence that maybe there's not different activity. But ultimately, we don't know.
All right, let's thank Kieran again.